say much.
All right. Thank you all for coming to the TSVP talk today. As you can see, Miguel Lurgi is here with us, and he will be talking about what we can learn from the Tangled bank, which is, a deep Darwinian reference for those of you unaware. Miguel comes to us from Swansea University in Wales, where he's been an associate professor leading the computational ecology lab for the past 6-ish years, I think. Before that, he was a postdoc doing postdocs in Australia, France, PhD in Barcelona, and an undergrad in Venezuela in mechanical engineering or some aspect of engineering. So he comes to the field of ecology through an engineering background. Miguel's work primarily concerns the analysis of the sort of topological and structural properties of networks, primarily species interaction networks. It's quite a hot topic in ecology right now, but it dates back to some of the founding work on ecological stability by Bob May. And as you can see, putting numbers to ecological networks is quite a challenge, and then analyzing them to understand sort of the drivers of population and community dynamics, is really an open field at this point. So I'm sure, with the rest of your time here in TSVP, if any of you are particularly inspired by this talk, please give Miguel an email. Just go to knock on his door, and he's open to collaborative opportunities from all of you, I'm sure. So with that, please begin. Thanks, Miguel.
Cool. Thank you very much for the kind introduction, Dave. Yeah. Actually, you said a lot of the things that I'm going to talk about, so probably I don't even have to give the talk. It's done. So, yeah, and as Dave said as well, I'm going to be here for 2 more months. Well, a little bit less than 2 months. So if any of you find any of this stuff interesting, please just feel free to come find me and we can have a chat about all of these things. I, of course, want to say thank you to OIST and the TSVP program for bringing me here. This has been an incredible opportunity, and I am really grateful for their support. Lynn and Jonas have been amazing at organizing everything, and it's incredible what they have going on here. It's a very nice thing to do. So yeah, as Dave was mentioning, this part of the title is a reference to a deep Darwinian thing, and that deep Darwinian thing is sorry, can't go through this. Is essentially this. This is a passage from The Origin, which is, at least in my view, one of the most influential scientific manuscripts in the last couple of centuries, I guess. And this is how basically Darwin ended his book. He said that it is interesting to contemplate the entangled bank, clothed with many plants of many kinds, with birds singing on the bushes, with various insects flitting about, and with worms crawling through the damp earth, and to reflect that these elaborately constructed forms, so different from each other, and dependent on each other in so complex a manner, have all been produced by laws acting around us. This is something that really resonates with me, because every time I see something like a new bird, a new animal, I cannot believe that it has been actually produced by the laws of physics, basically, right? So it's one of those things that really make you think about this. And importantly, one of the things that Darwin realized here is that all of these things kind of live together in an entangled bank of interactions of so many things. And this is kind of a recurrent thing that I want to talk about today, and how we have been approaching looking at ecosystems from this perspective, right? Of course, back in the day when he was writing these kind of things, he was not really already thinking about how to approach it, but there was this realization even back at the time where all of this was mostly natural history, right? And then after, going on a little bit more about summarizing the findings of the theory, then he ends by saying that there is grandeur in this view of life with its several powers, having been originally breathed into a few forms or into one, and that whilst the planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved, right? So basically, again, one of the things that, and of course, this is going to be my very personal and biased view of how we have come since Darwin's time until what we see right now, or what we know now based on different advances and how we are studying these systems. Basically, my hope is that I can sort of show you how I think about these things and how in my lab we have been trying to better understand these systems from ecological and evolutionary perspective, right? To try to use this entangled bank and not only characterize it, but try to understand it better, right? So back in the day, at this kind of around the same time when Darwin was writing The Origin, this other guy, who is another famous evolutionary biologist, Alfred Russel Wallace, he was trying to understand how species distribute along large spatial scales, basically. He's by many people considered like the father of biogeography, which is basically the mapping of species distributions. And one of his great contributions was the trace of this line here, which is called the Wallace's line. He realized while doing his work in the Southeast of Asia, across Borneo and Sulawesi, that essentially all the fauna that was found on the one side of on the left-hand side of that line was mostly related to like Asian origins, whereas all the fauna that was on the other side was most likely coming from Australasian origins, right? And these shaded areas, basically, which were not visible, you know, by the naked eye because this is covering water, then it's when they overlaid this on the extent of the continental shelf, they realized why this is happening. And the explanation for this was because these islands were connected during the interglacial periods, and these ones also likely, right? There is a part here that is kind of and was debated and still is. But the important thing about this is that people have been trying to keep understanding these patterns, and in particular, the kind of things that we as ecologists are interested in understanding is these kind of things. Like, why do we observe biodiversity most concentrated in some areas? And when you look at, in this case, this is the number of species across the latitudinal gradient, but not only the number of species, also when you look at the distribution of specific species, maps tend to look like this, or different fashions of these things. And these are the patterns that we are interested in understanding, right? So people started realizing these kind of things many years ago. But yeah, in these recent papers, they speak about these kind of things, right? So naturally, when you have patterns like this, there are many questions that sort of emerge, right? What do you want to understand from here? Why are there so many species together? How can they actually live all together, like in the same community? That was one of the main things, again, going back to the Tangled bank. Yeah, it was nice that Darwin realized that, but what are the mechanisms for that? How is that possible? Then there are some questions related to how environment interactions and evolution sort of together affect the structure of these communities. In particular, you know, like here we, for example, observe a very strong probably environmental gradient, and it is one of the proposed causes for this variation in diversity. And lastly, something that I'm going to talk also a little bit about towards the end is what are the implications of having this, you know, all of these species living together and their respective fluctuations in population dynamics for the stability and persistence of big ecosystems, right? Like how can this be like dynamically possible? So people started looking into these things, you know, back in the 1950s with the pioneering work by George Evans Hodgkinson. Hodgkinson basically proposed the idea of the niche as a way of understanding how species can share or not share if they competitively exclude each other a given space, right? So in a very simple example here, we have what is called the niche for two species in 2 dimensions. But obviously, when you extrapolate this to many more other needs of each species, this becomes a multidimensional hypercube of many things together. And that is what ecologists call the niche, right? So in this particular case, we focus on temperature and the size of food that species 1 and 2 can basically can live on. And here we can see that there are parts of the space, like, for example, this shaded area, in which S1 can live both because of food and temperature, but S2 cannot survive. So S1 is free to live here without any pressures from anyone. But as soon as you get to this part where S2 is also found because it has the right size of food, then this is where competition starts to happen, right? And this kind of things is the kind of thinking that we use to understand the interactions and the consequences of these interactions between species. How, because of these kind of things, species kind of competitively exclude each other and stuff like that. If you like me, by the way, if you are like me and you enjoy reading like classical papers about this, I cannot recommend this enough. This is one of Hutchinson's paper, Homage to Santa Rosalia, or why are there are so many species. This is a very nice kind of natural history introduction to all of these sort of things. So ever since then, you know, people started to think about what could be all of the dimensions of this niche, right? And one of the prevailing by now I guess it's a fact, is that environment is basically the main determinant of all these species distribution. These map things that we saw before is mostly because of environment. But then when you hone down into the more local scale, then biotic interactions become important. And a couple of years ago, we were looking into what are the trade-offs between these two things. Is it climate or is it diet, right? That drives the distribution, in this case, in host parasitoid networks of sawflies that live inside salix galls. So this is trees that have some parasites. The parasites form a gall in the leaves, and then there are parasitoids that go and use them for raising their eggs, right? And we had data from these communities in this latitudinal gradient from like the Mediterranean all the way to the top of Finland. And by analyzing the number of interactions that these species had and how many kind of habitats or environmental conditions they could live on, we found that indeed, you know, the occupancy or the distribution of these species is very much related to the climatic niche breadth, which means that they need a lot of the ones that have that can sustain more environmental conditions, they are going to find in many more places. That's, you know, very straightforward and intuitive. But one thing that was not like super surprising, but still nice to see, was that the occupancy was also very much determined by the total number of interactions, right? Which means that you not only need the right environmental conditions, but you also need to have the partners with which you interact, in this case, the resources for parasitizing, to basically increase your distribution, your geographical distribution. So now, you know, we kind of have an idea that environment and interactions, and you know that we kind of are building the sort of foundational blocks of why species live where they are and what are the main determinant factors. But one thing that we are interested in is to understand how these differences at the level of the species and their traits and their interactions and all that actually impacts the whole communities, right? And not only how they impact them in local scales, but also across spatial scales from local to global, or global to regional in some cases. So when you start thinking about these kind of things, one traditional line of work is to look at the different species and then count how many individuals there are for each of the species, and then try to analyze how the distribution of this or the relative abundances of each of these species changes, for example, in a geographical gradient, right? This is how people normally start looking into what are the consequences of different species for communities. But another sort of alternative, or not really alternative, but a complementary way of looking into this is what happens when we not only look at the proportions of the different abundances of the species, but we look at the how they are related to each other. So we convert objects that look like this, like a random sort of collection of species, into what we call ecological networks. Ecological networks are essentially a network in which nodes, which in the case of this picture, which is a simplified representation of the North Atlantic marine food web, are represented by these labels here, right? Those are the species, and then arrows going from one species to another represent trophic interaction, basically which species serves as a prey for a predator. And in this way, we can see how, for example, there are food chains going from the basal resources here, like phytoplankton, all the way up to black-legged kittiwake, right? So we use this sort of object to try to better understand the organization of communities and how this organization changes across spatial and temporal scales. So by doing this, we have, for example, found out what is the effect of, in this particular example, some work that we did a few years ago in the Pacific coast of Chile, looking at marine intertidal communities, mostly composed of invertebrates and algae along an intertidal gradient. And we build networks of interactions, of 3 different kinds of interactions, actually: trophic, positive as in mutualistic interactions, and negative as in competitive interactions, mostly for space in the rocks. But anyway, the details are not important. But we analyze these networks across this latitudinal gradient of environmental conditions. And as it turns out, in, like supporting also some previous hypotheses on the role of productivity on the shape of these networks, we found that more productive environments, in this case down here because of upwelling events from currents from the bottom of the ocean, make networks more diverse at the basal level, even though in higher latitudes they are a little bit more complex, right? I'm not going to go, by the way, I should have mentioned at the beginning, I'm not going to go through all the details in how we did each of these things, but if you're interested in specific papers or any of the examples that I talk about here, I, of course, will be more than happy to discuss the details that goes into these kind of things. So once we start understanding communities in this way, then we have kind of a pattern, a snapshot of something that we want to understand more mechanistically, right? So now we have a picture of how these ecosystems look like. But as I was saying at the beginning, one of the things we want to understand is how is this possible? What are the mechanisms that drive this, right? So some work that we have been doing to try to understand the scaling of biodiversity across space is based on foundational work that was started back in the 1960s, looking at how biodiversity scales across spatial scales. And in this seminal paper, The Canonical Distribution of Commonness and Rarity, Preston draw one of the, which with time turned out to be one of the most well-known patterns in ecology. Probably, I would dare even say that it's the most well-known pattern in ecology. So every ecologist has, at some point in their lives, seen this line. And this is essentially what is known as the species-area relationship. After Preston did this in the 60s, many people, for decades have replicated this exact same thing across many different taxonomic groups, across many different habitats, and across many different environments and parts of the globe. Basically, what we are looking at here is how the log number of species scales with the log size of the area that you sample, right? And interestingly enough, these lines always look like this. They always follow this shape, which is a scale-free or power law relationship between the number of species S and area, which is power to some exponent. Obviously, because this is ecology and we never have exact answers to everything, the exact value of this set is, as you can imagine, the subject of very heated debates, and I am not going to go into that. But essentially, there is some set that applies arguably to many systems and possibly universally, right? So we wanted to start from the basics, and this was kind of the beginning of that. And we wanted to start from the theory that basically kind of tries to give an explanation of that, of this pattern. And that theory is called the theory of island biogeography, that was proposed by Wilson and MacArthur back in 1963. And basically what they say is that the number of species that you find on this case on an island is a dynamic equilibrium between the colonization, which is these orange lines that you see here, and the extinction of species, right? For those of you who are more mathematically inclined, this is the exact model for that, right? Basically what it says is that the rate of change of the number of species is a function of the colonization multiplied by how many species are left in some pool. So SP here is the pool of all possible species, and S is the actual number of species that you have, minus the extinction of the probability of or the rate of extinctions of those current species. I'm not going to go through all the details in this plot here, but basically these two lines mean that if you consider islands that are far or close to this pool and islands that are larger or smaller, and this is where the area comes into play, basically that sort of modulates how much colonization and how much extinction you have. And where these lines cross is where you have the equilibrium number of species that you will expect for that thing, right? So if you solve this equation to equilibrium, so making this equal to 0, you get that you can calculate the number of species only by having these two parameters, C and E, right? And this is where these lines meet here. So basically what we wanted this has been known for a while, right? And people, have kept trying to better understand these patterns of species area relationships, but also many other things. And what we wanted to do was to say, well, now we have a very sort of decent understanding of this thing here. We want to basically better understand how networks of species interactions also scale with area, right? And in 2018, we came up with a theory that kind of tried to explain this, right? Basically, we came up with 3 different models that were based on previous work by other people. And we tested the differences between these 3 theories of potentially how area or how the structure of interaction networks scales with area. So in the first model, we basically took the species area relationship as it was, and then we sampled randomly the number of species that you would expect from a given area, and we kept increasing the area until the sampling was complete. And by doing this random species selection, we built networks that contain that number of species. And that gave us some shape of a species area relationship, right? So that would be kind of the baseline expectation based on the species area relationship as explained by MacArthur and Wilson. And this is how, we have this kind of baseline expectation. Then our second theoretical model was more about, instead of sampling number of species randomly, let's have a parameter that is that is a proxy for the size of the area, which is colonization divided by extinction, right? So what we did was we simulated the colonization extinction dynamics of each of the species within that network. And then by changing this value here, we built networks at different areas and which were of different sizes to see what is the effect of, instead of choosing species randomly, we assume colonization extinction dynamics and how that affects the scaling of this of the shapes of these networks, right? So surely enough, we saw some differences here. This is the differences across the different models. Again, I'm not going to go through the specific details of what each color means here, but the important thing is that the blue line, the red line, and the orange line are different, right? So these were the expectations from all the different models that we came up with, not only in terms of area and the number of links per species, but also, for example, in the relationship between species and number of links and some other things, right? So these were kind of the expectations of our theoretical framework. The next step was then to try to understand whether this actually happens in nature, right? So we went out to the field, well, not the field, we went out to the internet and we found a lot of information for how species are distributed across Europe. And we found a very high resolution network or food web of interactions between all of the tetrapods in all of these locations. And we built single networks at the level of the grid in 10 by 10. We basically split Europe in 10 by 10 grid cells, and we built networks for each one of these locations based on the species that live there and based on this meta web of many interactions, right? And we built the exact same relationships that we expected from our theoretical models, but from empirical data. And as you can see here, some lines kind of look similar to what we expected from our theoretical model, but others do not. And this is where we start making hypotheses about what are the other mechanisms that we did not consider that are also adding to these things, right? By the way, the different colors here are different biogeographical regions, because as you can see here, it turns out that environment, so the type of habitat that you draw these network area relationships on, it's a very, it seems to be driving a lot of this variability, right? And similarly, for the number of species and the number of links, we got a scaling that is kind of in agreement with what we expected theoretically, but it was nice to confirm that, basically. Cool. So one other thing that we are interested in is because, nowadays my students are very interested in conservation, right? And well, also myself, but, like when you are very eager to make a change and, you really want to contribute to science, you want to save a species, right? That is the thing when you are an ecologist. You want to be able to provide answers to, the global biodiversity crisis, right? So one of the things that we are working on is how we can use this type of knowledge to better inform conservation, right? Or to at least try to understand how conservation is actually good for ecosystems. So if we look at the, So if we look at the two relationships that we obtained from our kind of like species or network area framework, we have that, the slope at which the number of species scales with area is very different from the slope with which the number of links, the red line here, scales with area, right? Which means that ecological interactions are behaving differently than the number of species when area changes, right? And this is very important because biodiversity is not only about species. Biodiversity is a bunch of many things, including ecological interactions. So the expectation from when you reduce area, which is a common thing that happens every day when we lose habitat in many different ecosystems, is that the number of species that you will lose is not comparable with the number of ecological interactions that you will lose. So using this framework to think about this, we would expect that you would lose more ecological interactions than species, right? If this is true, right? By the way, if you are interested in kind of this of these sort of things, there is I am currently working on a chapter that will be published in August on an edited book, Biotic Interactions Across Gradients and Scales, in which we actually talk about all of these things and how we can use the this knowledge for understanding ecosystems. So using this kind of framework and thinking about this, we wanted to better understand what is the effect of area and some other factors on the changes in network structure. And for that, we wanted to look at the differences in network structure or food webs across protected areas in Europe, right? The reason why we did it in Europe is because it's where we had the data, right? Specifically the interaction network data. So this is how that network of trophic interactions between tetrapods in Europe looks like from this paper here. And what we did was to try to analyze what is the effect of protected areas on the network of trophic interactions between these species. So what we did was we built networks at the level of the cell in each of these green and orange areas. So the green areas are the protected areas in Europe, and the orange areas are non-protected buffers around those areas. And we wanted to basically compare the networks inside of the protected area with the outside, right? And to do this comparison, basically, when people study networks, what they usually do is they quantify a bunch of properties that are relevant for network structure. Like, for example, connectivity, modularity, all sort of things. Again, I'm not going to go into the details of each of these things, but if you're interested, I'm more than happy to chat about that. And then we related the differences between the structural properties of networks inside and outside with some environmental variables, right? And two of the variables that actually came up as very significant or very important in determining whether protected areas are good for biodiversity were the diversity of land cover types and what we call protected area continuity, but it's basically a measure of fragmentation, right? So we are in that part of the graph where you are losing habitats because the habitat is becoming more fragmented. And that means that you are losing parts in the middle of the habitat. So the protected areas are not a single big thing, but they are like small patches that are somewhat connected in some way to each other. And we can see here that, for example, the mean body size of top predators was much better or much higher when land cover types was, when the habitat was more heterogeneous, essentially. And similarly, when you had more intact protected areas, the number of intermediate species was smaller, which means that, again, you are favoring large species. In food webs, by the way, favoring large species is a good thing because it means that you have a healthy enough base of your food web to maintain large things, right? Like, for example, if you don't have enough zebras, you are not going to have lions in your habitat, right? This is the kind of thinking. That's why, even though this line is going down here, it's actually a good thing because it means that intermediate species are less because you now have big things at the top. Cool. So this is, until now we have been speaking about structural properties of the network. So in our minds, networks are still these kind of static things, right? You have these nodes, you have these links between the nodes, and then you quantify things. And we have seen how they change across environmental gradients and how they change across habitat degradation, right? But one thing is that is kind of very relevant and that goes into the consequences of having these different structures is looking at what does it mean for community persistence, right? Like, what does it mean to have some specific type of network instead of another one for the persistence of all of those, species living together that we were talking about at the beginning, right? And the understanding of this relationship between structure and persistence or stability goes back to the pioneering work by Robert May in 1973, in which he was using random matrix theory, basically taking random matrices like this and using what in this kind of approaches is called linear stability analysis. basically looking at the eigenvalues and eigenvectors of the Jacobian matrix for the community. And he came up with this relationship, which is, again, one of the main tenets in theoretical ecology ever since the 70s, which says essentially that as complexity of the community increases, then the chances of that community to persist are smaller, basically. And in particular, is if the square root of the product between the number of species and the connectivity of the network multiplied by the mean interaction strength between the species in that community is larger than 1, larger or equal than 1, then you should expect the collapse of your ecosystem. And surely enough, these kind of things, which may identify theoretically back in the day, have been actually shown in the lab, right? So this is data from microcosms from this work here by Fox and Mike-Steed. And you can see how, as complexity increases, the probability of feasibility of their microcosm communities basically follows this sort of shape, right? And even though there have been some tests of this and they look like this, in general, ecosystems are very much more complex, right? So we go outside and we see systems that are way more complex in terms of number of species and interactions than what May imagined in his random matrices. So, and obviously he was aware of this, right? So when he realized this, he sort of put forward this sort of challenge, right, for theoretical ecologists and ecologists in general. He said that the challenge, based on what he found, is to identify the devious strategies that a natural system uses to increase species and connectivity capacity without sacrificing stability and persistence, right? So, and ever since then, essentially what ecologists, including myself, we have been doing I haven't been doing it for decades, but people have been doing it for decades. They have been trying to identify these devious strategies, right? And it turns out that some of these devious strategies are essentially the shape or the way in which the interactions between species are distributed in the network, right? So if you know a little bit about networks, not only in ecological networks, but networks in any context, you might know that networks across many domains have very kind of universal characteristics. One of them is the power law distribution of their interactions, right? So the number of links is distributed in a power law way, in which there are many species that the majority of species have very few links, and only a few of them are very central. And these kind of things have implications for like fault tolerance and stability and so on, right? So these kind of like very fine-grained structural properties turn out to be these devious strategies. And again, people have tried to show which ones are more relevant. Modularity, for example, has come up as one important one, and so on. And working towards this, we have been trying to basically identify which ones of these structural properties are relevant to ensure a very specific type of stability in food webs, right? And in particular, the robustness to invasions. Like what makes a network more robust, or an ecosystem robust to an invasion, and what it makes it so susceptible, right? So for that, we did some theoretical work. I am, again, not going to go into the details of each of these equations. I am just putting them here in case anyone in the audience is interested in these kind of things. But basically, we model food webs that look kind of like this, in which each of the balls here are different species, and the green things are interactions between them. And we use a generalized Lotka-Volterra approach, in which the things that you see in here, some of these terms are actually allometrically scaled, right? So this is relevant because it makes the modeling of the food web more realistic. But the important thing is that we are modeling the dynamics of each these species and how they persist when they are together with other species in the network. So we use this equation to simulate dynamics that look kind of like this, right? So this is the population dynamics that come out of that model. And in our experiments, in our in silico experiments, we basically waited for some time until the transient dynamics of that system passed and the system was stable. And at that point, we introduced a new species, which is this green point here, right? Before the introduction, we measured a bunch of network properties. Again, I'm not going to go through the details of each of these, but basically, this is how network ecologists quantify the shape of that network, right? And then after the invasion event, we let the community to reorganize after that kind of disturbance. And we analyzed the shape of the network after that, right? And that obviously allowed us to compare this with this, but also importantly, what were the properties of many different of these guys here that enhanced or prevented the invasion by the green node, right? And surely enough, we found some of these devious strategies. So some species, some network properties, like for example, connectance, the number of mean food chains, or the diet breadth of the resident species are very good predictors on the stability of that network or the robustness of those networks to invasions, right? And another thing we looked at was how the stability of the populations. In here on the x-axis, I have the time after successful invasion, and on the y-axis, I have the proportion of stable networks, right? And we can see how, depending on the connectance or the connectivity of the network, so the networks were more or less stable, right? So more connected networks were more stable in terms of population biomass, even though they were less stable in terms of community biomass after the invasion of the species, right? So which means, again, that after a perturbation by some external stress on the network, then the network properties are very important to determine whether that community will be more or less stable. Cool. So now we know a lot about the ecological consequences of this network structure and some of the mechanisms that may give rise to them. But I started the talk talking about Darwin, right? And for Darwin, everything was about evolution. In one of the previous talks here, we also saw that nothing in general, but in biology, makes sense except in the light of evolution, right? So what we are trying to do now is to try to understand what is the role of evolutionary processes into the assembly of these communities. And for that, we are looking at bacterial communities that live inside multicellular hosts. This is marine sponges in particular. Marine sponges have a very kind of interesting sort of life history strategy, which is that they can do either of two things. They can either pump a lot of water and try to get many nutrients from the water, including eating microbes from the environment. And there is another strategy in which they keep good microbes and they basically exploit them for resources, right? So we took empirical data from microbial communities inside marine sponges from essentially all the seas around the globe. And we analyzed the community structure of each of these things inside the marine sponges. The community structure, this is just for you to have a picture of how this looks like. But in here, we have on the x-axis all the different sponge species. And on the y-axis, we have all the different bacterial phyla that you can find. And the color here represents the relative abundance in each one of them, right? So again, we are not going to understand everything in this picture now, but the important thing from here is that different species basically like different microbes, right? And we wanted to understand whether the way in which they like these microbes is determined by either environmental or evolutionary features, right? So what we did was we used those microbes inside the sponges, and based on which species they were most commonly found on, we built networks of host microbe interactions, right? The big nodes here are sponge species and the small nodes are microbes. So the network kind of look like this. And we analyze different structural properties of this network. One very important structural property, which is shown in this picture, is modularity. Modularity is the extent to which interactions within the same group of nodes are more common than with nodes outside of that module, right? Using this information, we try to understand what was the relationship of this structural property of the network, of this modularity, to the evolutionary histories of the different hosts, right? And surely enough, we found this very beautiful phylogenetic signal in which we can see that all of the high microbial abundance species, so those species that I told you that like keeping their microbes rather than pumping them through the water, they like basically exploiting the microbes to get their own food, they all kind of cluster together in this branch of the phylogenetic tree, right? Which means that they have this, we discovered this kind of very intimate coevolutionary relationship through time in the way in which the modules of that network are structured, right? So there is a phylogenetic fingerprint on how this network is basically assembled, right? So to follow up in this kind of thinking, we are now working on trying to understand more theoretically what are the bases of this. Because again, this is the pattern, right? We see that there is a relationship between these things, but we want to understand what are the potential mechanisms behind that. And to do that, we are trying to develop some theory on what are the ecoevolutionary drivers of assembly of complex communities with multiple interaction types. For that, we set up a theoretical model that, as a cartoon representation, kind of looks like this. So we start with a small community, in this case of 3 species. And then we create what we call these assembly events, in which we take one of the species in the community and we make it a parent species. And the offspring species is introduced based on some mutations from the interactions with the parent. And then after we run the dynamics of the microbial community again, and after some extinctions are happening or not, then another sum of new equilibrium is reached. After that, we introduce another species using that same evolutionary algorithm, and so on until we reach very, very large communities, right? The way in which we simulate these dynamics is, again, using a generalized Lotka-Volterra equation. I'm not going to go into the details of everything here, but basically, essentially, the important things are that you have some reproductive rate, you have some competition between different species, and all these terms here are the different interaction types that we can have. Because the difference between this model and the one I showed before is that in the other one, we only have consumer resource dynamics. There are only predators and prey, whereas in here, we can have any type of interaction, from amensalism and commensalism all the way to mutualistic interactions. So not only we are changing the links between the species, so the structural property of the network, but we are changing these parameters here, which determine how strongly they interact with one another. Again, I don't have the time to go over all the details of here, but if you are interested in these kind of things, more than happy to talk about this. This will actually should come, I hope, next week, if they let me pay, but in Nature Communications. But essentially, one of the things that we found is that basically there is a very important trade-off between the harvesting cost, so how much it costs you to have a positive interaction, how much it you need, how many resources you need to devote to actually be able to extract benefits from a resource, and the interaction strength that you have with that resource, right? So. And basically what this trade-off means is that there is a very interesting threshold here under which the proportion of mutualistic interactions, it's noticeably higher only when the strength of the interactions are high, right? So you will we only expect ecosystems to develop mutualistic interactions if those interactions are very, very beneficial, and in particular, if they are more beneficial than what it would cost for the partner to invest in that interaction, right? And conversely, if the other thing happens, then we would expect communities to have a predominance of competitive interactions, right? So there is we found this kind of change in regime that is dictated by the trade-off between these two parameters. And we wanted to see if there were, any fingerprints of these kind of things in nature. So for that, we looked at the community composition of microbial communities inside of the gut microbiome in humans. And this is what I'm showing here in the black dots. So these black dots are actually empirical data from gut microbiome, and the green dots here are the outcomes of our model. And in here, we see two macroecological patterns that are commonly used to understand gut microbiome data, which is the average. Basically, this is the abundance distribution of the species as the relative abundance. And you can see that there are many species, many species that are relatively rare and a few that are super abundant. And our model is able to recreate this pattern. And then if we look at the correlation coefficient between these two species, so how often two species are high in abundance together and how often they are not, we see that our model is very good at predicting the negative correlations, so basically the negative interactions between the species. And we have a little bit of trouble adjusting this. So we are now trying to think about what are the further mechanisms that might not be evolutionary, or maybe yes, but in a different way, that actually can make this sort of matching better, right? But the important thing is that it looks like this kind of eco-evolutionary thinking can give us some clues on the structure of the gut microbiome. Cool. So yeah, I don't really have much more time left, and I think that's it from me anyway. But yeah, I just want to say that obviously all the things that I was talking about to you today, have been done by all of my postdocs and PhD students. So without them, none of this would have been possible. And some of my collaborators that I am currently working on with in different things, from intertidal to microbial communities. And of course, my funders, the Leverhulme Trust has funded much of the microbial work. And again, thank you to the TSVP for funding my stay here. Thank you very much.
All right. Thank you, Miguel. So we have time for some questions. Just please raise your hand and shout out the questions. We have a active microphone in the ceiling, I guess.
Sorry, I forgot that I had to say one more thing. If anyone is interested in these kind of things from a more kind of like, basic perspective, we are not going to go into complex models of that. I am going to be running this mini course on the week of the 17th of March. It's only 3 sessions, and I figured that I wouldn't make it too long or complex because, I wasn't aware of the level of the audience. But yeah, if we want to add more things, I'll happy to do. But basically, we're going to touch upon things from single species population dynamics to networks and stuff like that. So if there are any students here interested in that, just please come and join us. Yeah. Now I can answer questions.
Any questions?
Sam.
Am I just shouting?
Yeah, just shout.
Okay. Thanks, Miguel. That was really cool. I've got a question about the Nature Comms paper at the end there, the heat maps that you were showing. I had a bit of a hard time getting my head around this, but the complexity seems to be driven much more by the connectance than by the richness. Is that fair to say? Like those two look like they map almost one-to-one.
Yeah.
Can you break down a bit what that means for me? Because I couldn't quite wrap my head around what that means kind of ecologically.
Well, it means that when you have more species, it seems like that you can only have it when you have less interactions, right? So you have a species, which again, I guess that it matches sort of our expectations from previous work as well, right? If you have more species, and going back also to May's thing, right? You have,
yeah. Okay, thanks. Yeah, I figured it.
Yes.
So in the paper, you presented a lot of papers on. It was on spatial the linkage area relationship.
Oh yeah.
Way back, yes.
Yeah.
Okay, so a lot of the sort of mechanism underlying the species area relationship. I mean, area is sort of just a proxy for habitat heterogeneity, right? And so if you have a highly heterogeneous habitat, that is presumed to be what's really affecting that Z value, okay? If you have a more or less heterogeneous habitat underlying that area, are you expecting to see higher amounts of modularity in those food webs, or did you look at modularity? Because wouldn't you assume that would be the case? And if that's the case, then your results with showing how modularity is actually like really related to the success or non-success of invasion might actually play out if you were to look at like separate invasive species out in this.
Yeah.
Sorry, a complicated question.
So basically to try to account for this habitat heterogeneity, but because obviously, habitat heterogeneity is bound to change that slope in the species area, not only the species area, but also the network area. But that's basically what we tried to account for by doing this. So we wanted to split the map into things that were of the same habitat, right? But obviously within biogeographic regions, you still have very diverse habitats. And what we are saying is that very likely these kind of lines here are because we are still getting more habitats into the mix, right? And that's the in terms of modularity, we didn't really look at modularity in these like empirical examples. We looked at modularity in the model, and as expected, because we don't have a habitat heterogeneity, it looks flat, right? So there wasn't a lot of sort of effect of modularity on the theoretical sort of framework, which makes sense because we don't have habitat heterogeneity. But I would expect the modularity to change when you add more habitats. That's correct. And in terms of what that would mean for invasions, I think that I don't know how to think about that because I haven't really thought about that, but because the invasion part is kind of independent from this work. We didn't do it with this data set. So it might be the case that when you consider ecosystems that have more habitats, it will be harder to invade like the entire network, but you will possibly be able to invade specific modules.
It kind of, I just wonder, because there's that paradox that species richness predicts negative like sort of hindrance to invasion at small spatial scales, but at large spatial scales, its invasion success is positively correlated with species richness. And so I'm just wondering how that might map onto like food web.
I wasn't aware of that result. Okay, that makes sense. I
won't take up too much more time.
Thank you, Miguel. Also for me, some parts were very interesting. And one question, you showed us this article from Hutchinson from 1959 with the question, why are there so many kinds of animals? Is that still the main question today?
I guess that to some extent, yes, we have gone a long way into understanding, like part of the answer to that question is the structural properties of the ecosystems. And that is also why in current conservation, like discussions or debates, people are very aware that losing some specific species will produce a collapse, because there are one of the reasons that people think there are so many species is because there are some of them that are like gluing the whole thing together, and this goes back to the for example the power law shape of degree distributions in networks in general, right? These kind of things. So I think that we have gone quite a long way into understanding why there might be so many types of animals. And now it's more about how we can use that knowledge to actually try to not lose those animals, basically. I think so, yeah. But again, this is my opinion. And, if you ask this to another ecologist, they might answer differently, yeah. Yeah, that's.
Yeah, I have a follow-up on both of the previous questions, and it's also about modeling. So an ecosystem maybe starts from bacteria and it ends with large animals and large trees, and there's a lot. But as a scientist, I guess you must make choices to do the modeling. And so you take some species into account and you go. Now, how do you know that your results actually are not sensitive to the choices you make in species you take into account? So there's, as I learned, there's scale dependence. And also, very naively, if there's a nuclear war, then the bacteria, maybe the ants, will survive, but we won't. So the scale dependence to external circumstances seems enormous. So how do you make your choices?
Yeah, I guess that is a very hard question to answer. Well, one thing that I should mention is that for some of the stuff that I spoke about, the whole thing is purely theoretical. So we didn't make any like specific choices about any particular system. Like, for example, the invasion thing, it's like a general expectation for any sort of networks that looks like the ones that we use, but we didn't use like specific choices for a particular ecosystem, right? When thinking about choices for a specific system, like right now we are doing, for example, some work in understanding the evolutionary origins of these high versus low microbial abundance sponges that I was talking about. And to understand that, we have made very specific choices about that specific system. And obviously the predictions from those models are bound to be only relevant for that specific species, right? So if you try to apply that knowledge to any other type of host microbiome community, it's probably not going to hold, right? So I think that the difference between ecology and physics in that sense is it's very large, because in physics, I think that as far as I understand, I don't know much about physics, but people try to make models that are like general to everything, right? In ecology, that happens sometimes in for example, those cases that I mentioned where the thing is purely theoretical. But usually when we try to model a specific system, the choices that you make, especially for the mechanisms that you put into the model and also the parameter values that you use for them, are usually only relevant for that system. So yeah,
it's more like history than like physics in a way.
Yeah, I guess. Yeah.
One more question. We have time. Going once, going twice. Okay, let's give Miguel another hand.
Thank you.